Welcome to the Visual Histology videotape series. In one of the past programs, we've described cells, and now we're moving to tissues, a more complex level of organization. The tissue we're going to be talking about today is epithelial tissue, and it's really extremely interesting. We're multicellular organisms, and consequently our functions are compartmentalized into different organs, and epithelia are the tissues that line or cover internal and external surfaces, organs, blood vessels, our whole body. They're extremely important. Now, since we're biologists, of course, we have to classify them because they come in a number of different sizes and shapes, and very often students shudder at the thought of classification. There are more names to learn, but epithelium is very easy to learn to classify. In the first place, epithelia consist either of a single layer of cells, in which case they're called simple epithelia, or many layers of cells, in which case they're called stratified epithelia. Now, the shapes of the cells that make up these epithelia vary. They can either be flat, in which case they're called squamous cells, or they can be just about as tall as they are wide, in which case they're called cuboidal cells, or they can be taller than they are wide, in which case they're called columnar cells. Consequently, if you have a layer of cells that's single and flat, it's a simple squamous epithelium. If you have a single layer of cuboidal cells, it's a simple cuboidal epithelium. If you have a single layer of columnar cells, it's a simple columnar epithelium. Now, it gets a little more complicated with the multilayered epithelia. If you have an epithelium that consists of more than one cell layer in thickness, but the upper layer is flat, it's called a stratified squamous epithelium, because the adjective squamous refers to the shape of the uppermost layer of cells. Consequently, if you have an epithelium that's more than one cell layer thick, but the uppermost layer is either cuboidal or columnar, it's called a stratified columnar epithelium. Now, most epithelia that you'll encounter fall into one of these categories, but there are several that don't. For example, there's a kind of epithelium, like the epithelium that lines the trachea or the upper airways, that looks stratified, but it is not. It's really a simple layer. This is called a pseudo-stratified epithelium. And the epithelium that lines the bladder can look different depending upon whether the bladder is full or empty. Its appearance undergoes transitions. It's called transitional epithelia. But I think you'll see all these are quite easy to recognize when you look at them with the microscope, so let's go to the laboratory and have a look with the microscope and look at the histology of epithelial tissue. The first kind of epithelium we're going to look at is simple squamous epithelium, and that consists of a single layer of flat cells. This image right here is a section through the Bowman's capsule of the nephron of the kidney, and it's lined by an epithelium, which is indicated by the arrow, which is a simple squamous epithelium. The epithelium here is extremely thin, extremely flat, and what you can see are these little purple bulges. Each of these represents the nucleus of a single cell. So there's a nucleus here, and then a string of cytoplasm here, and a nucleus here. We can see this a little bit better if we go to higher magnification, so we'll switch over to another image where we can look at it where everything will appear larger. This is a higher magnification view of the simple squamous epithelium that lines the Bowman's capsule of the nephron of the kidney. The arrow here is pointing to a single nucleus. The cytoplasm of the cell is just a thin strand that extends over here to this next pair of nuclei. This section has been stained by a particular stain called Masson's trichrome, and it's very nice because it will stain the nuclei of cells purple, it'll stain the cytoplasm red, and it will stain connective tissue green. So this green line here represents the basement membrane, the connective tissue, upon which this simple squamous epithelium sits. There's another kind of simple squamous epithelium in here. It's the epithelium that lines this bundle of capillaries, the glomerulus, which is in the middle of Bowman's capsule. These cells are so thin and flat, so squamous, that you can hardly see them. Their cytoplasm just looks like a hair. But here's the nucleus of one of these epithelial cells, and here's its cytoplasm. In this particular section, there's another kind of epithelium. There's a simple cuboidal epithelium that lines the proximal convoluted tubule of the nephron. The simple cuboidal epithelium is a series of cells that are roughly isodiametric that are aligned right next to each other. So instead of being flat, as in simple squamous epithelium, they are cuboidal, as their name suggests. We'll look at another example of simple cuboidal epithelium in the next image. This image is a conventional section stained with H&E through a follicle in the human thyroid gland. The functional unit of the thyroid gland is the follicle, and the follicle is basically a sphere of this purple material here called colloid that's surrounded by a ball of cells. The ball of cells is a simple cuboidal epithelium. Basically, it's a sheet of simple cuboidal epithelium that secretes this colloid, appears to be wrapped around it. And again, the simple cuboidal epithelium consists of a row of cells that are roughly as wide as they are high, that are spread in a monolayer. All of the cells contact the basement membrane. There's one thing that's a bit problematic with the nomenclature, and that is the following. If you look carefully, some of the cells, like these, look rather flat, and some of these down here look rather tall, and yet it's all defined as a simple cuboidal epithelium. Histologists love to name things. Many things have two or more names. This epithelium right here would be called a low cuboidal epithelium. This epithelium right here would be a high cuboidal epithelium. Now let's have a look at this same epithelium at higher magnification in the next image. This is a higher magnification image of the simple cuboidal epithelium that lines the follicles of the thyroid gland. The arrow is pointing to the surface of the epithelium, and the surface contacts the colloid, which is the pink material in here. Right underneath the epithelium is a capillary bed. You can see a capillary running along here, and you can see the individual red blood cells here and here. This simple cuboidal epithelium has a row of nuclei which are quite prominent. These nuclei appear as oblate spheroids. They're purple. The cytoplasm is pinkish. And one problem is that it's very hard to see the boundaries between cells. You can sort of sense where one is here, but you really need the electron microscope to distinguish the cellular boundaries where one cell meets another. These cells manufacture the colloid, which is in the colloid space here. When they're active, they tend to be quite tall. When they're inactive, they tend to be quite flat. Let's go now to the electron microscope and have a look at an electron image of the simple cuboidal epithelium that lines the thyroid gland. This is the simple cuboidal epithelium that lines the follicle of the thyroid gland as seen by electron microscopy. The arrow is in the lumen of the follicle, which is filled with colloid. The colloid is the amorphous gray substance up here. The surface of the epithelium is here. The base of the epithelium is here, and at this magnification you can see the basement membrane right here, this sort of filamentous wavy line upon which the epithelium rests. This is the nucleus of one of the epithelial cells. This is the nucleus of another cell. These two cells here and here are neighbors, and you can see their point of contact is represented by this dark line here. And this dark line is created by the juxtaposition of the two cell membranes that are opposed against one another. So in the electron micrograph you can see cell boundaries very clearly because you can see cell membranes. In the light microscope you can't. Now we're going to turn our attention to the simple columnar epithelium. A simple columnar epithelium is a single layer of cells, and those cells are taller than they are wide. They're shaped like columns, hence the name. This image you see here is a particularly good example of simple columnar epithelium. This is the epithelium that lines the surface of the intestine, and this piece of tissue in particular is an H&E stained longitudinal section through an intestinal villus. Right here the arrow is pointing at the base of the epithelium. The surface of the epithelium is up here. It's covered by a so-called brush border, which is a group of tightly packed microvilli. The detail in this section is quite good. You can see little filamentous bodies here. These are mitochondria, which are up in the apical end of the cell. The nuclei of the cells are here, here, and here, and just by inspection you can see that this is a simple columnar epithelium. Like all epithelia, it rests upon a basement membrane which is located here, but in this case the basement membrane is too thin to see, so you can't see a clearly demarcated basement membrane. The basement membrane rests upon some loose connective tissue. In this case it's called the lamina propria. It has all different kinds of cells in it, and is underlain by a large dilated capillary here. We're going to look at a different kind of epithelium now, the so-called stratified squamous epithelium. And stratified squamous epithelium is, first of all, a stratified epithelium, which means it is many cells thick. And second of all, the cells that comprise the uppermost layers are flat. Stratified epithelia are always named by the shape of the uppermost layer of cells, not by the general shape of the cells that are found throughout the epithelium. This particular section is an H&E stained section through the esophagus. It's a cross section, and the epithelium of the esophagus extends from here to here. The surface of the epithelium is here. It contacts the lumen of the esophagus, which is up here. And as you can see in this low magnification image, the epithelium is many cell layers thick. The bottom layer of cells here looks purple, and that's because the nuclei are all lined up and the nuclei are purple. The bottom layer of cells undergo repeated mitotic divisions and give rise to other cells that migrate through the epithelium and become flatter and flatter as they reach the surface. Eventually they're shed into the surface here as they're worn out, and they're replaced by the mitotic activity of the basal layer of cells, which is located down here. We can see these details a little bit better at higher magnification, so let's look at a higher magnification image of the same specimen. This is a higher magnification view of the esophageal epithelium. The arrow is at the junction of the epithelium and the connective tissue upon which it rests. So the arrow points to the base of the epithelium, and the full thickness of the epithelium extends from here up to the surface. Now, this is not only a stratified squamous epithelium, but it is also called a stratified squamous non-keratinized. And the reason for that is that all of the cells have nuclei that you can see in them. And the nuclei are present even in the uppermost cells, such as this one right here. So when you have a stratified squamous epithelium and you can see nuclei in the uppermost cells, it's defined as a stratified squamous non-keratinized epithelium. There is also a kind of epithelium called stratified squamous keratinized epithelium, and we will look at that in the next image. This is a light microscopic image of a very good example of stratified squamous keratinized epithelium. Here the epithelium is extremely thick. The arrow is pointing to the surface of the epithelium here. The outside world would be up here, and the base of the epithelium is all the way down here. The base of the epithelium is purple, and the pink material upon which it rests is connective tissue, and this tissue sample is palm skin from the monkey. The skin is built to withstand a considerable amount of abrasion. Consequently, it's covered by a very thick horny layer, a keratinized layer, which is called the stratum corneum of the epidermis, which extends from here to here. Now, the significant thing about this tissue is that these cells are completely dead. They have no nuclei left in them. They're keratinized. They're completely filled with keratin. So from here to here is the keratinized portion of the stratified squamous keratinized epithelium, and then the living cells extend from here on down. All of the layers will be covered later in the videotape on the skin. We're going to look at a different kind of epithelium now, a very unusual kind of epithelium. This is the so-called stratified columnar epithelium. The arrow is here in the lumen of a duct. This duct is the excretory duct of a salivary gland, and the excretory ducts of salivary glands are one of the few places in which you find this unusual type of stratified columnar epithelium. The surface of the epithelium is here. It extends down to here, and at this low magnification it sort of appears as a bunch of purple nuclei that are stacked on top of one another. To get a real feeling for the nature of this epithelium, let's look at it at higher magnification on the next image. This is the higher magnification view of the stratified columnar epithelium that lines the excretory ducts of the salivary gland. The arrow is at the base of the epithelium. The epithelium, again, is supported by connective tissue, which appears pink here, and all of these purple bodies are the nuclei of the cells that comprise the stratified columnar epithelium. As you can see, these cells are stacked on top of one another. The surface of the epithelium is right here, and the lumen of the duct that's surrounded by the epithelium is right here. The next kind of epithelium we're going to look at is pseudostratified columnar epithelium. This is a light microscopic image of an H&E stained section through the trachea of the monkey. And it's a very interesting section because it shows a very good example of pseudostratified columnar epithelium. In fact, to be precise, this is a pseudostratified columnar ciliated epithelium. The cilia can be seen here at the epithelial surface. They look like little brush bristles. They're very small. Each one is about 0.2 of a micrometer thick, which places them right at the level of resolution of the light microscope. These cilia serve to move mucus along the surface of this epithelium. The mucus is secreted by these large goblet cells like this one here. The cell next to this goblet cell is a ciliated cell, so you have two different kinds of cells here. One kind produces mucus, the other kind has cilia that move it along, and their nuclei are at different levels. There's a third kind of cell in this epithelium, the so-called basal cell, like this one here. Basal cells are small cells which are right next to the basement membrane. The basement membrane is visible in this preparation. It's quite thick. It's this light pink line right here. The basement membrane extends from here to here. The whole epithelium rests on top of that basement membrane, and each of the cells is in contact with the basement membrane. So although the nuclei appear to be at different levels, like this one here, and this one here, and this one here, giving the epithelium an appearance of stratification, all of these cells are in contact with the basement membrane. So the epithelium is not stratified, it is simple. But since it looks stratified and isn't, it's called a pseudostratified columnar epithelium. The microanatomy of the ciliated pseudostratified columnar epithelium is considerably clarified by electron microscopy. This is an electron micrograph of a thin section taken through the bronchiole, and the arrow points to cilia, which are on the surface of the epithelium. This is one of the ciliated epithelial cells here, and this is its nucleus. This is the tip of a mucus-secreting goblet cell up here, and another mucus-secreting goblet cell can be seen here. Down at the base of the epithelium, you see these small, dense basal cells here, here, and here. These cells, by mitotic division, give rise to replacements for worn-out ciliated cells and goblet cells. When you look at this micrograph, it's apparent that all of the cells do make contact with the basement membrane. Consequently, it's not truly a stratified epithelium, but rather a pseudostratified columnar ciliated epithelium. This is the last kind of epithelium that we're going to discuss today. This is the so-called transitional epithelium. This particular section here is a section through the bladder of the monkey. The arrow points to the base of the epithelium that lines the bladder. The epithelium consists of cells whose nuclei are reddish and the cytoplasm is pink. This is stained with a Mallory's trichrome stain, in which the cells are red to pink, and the connective tissue bed on which the epithelium lies is stained blue. So this is the base of the epithelium right here, and when you look at it, it looks like it's a stratified epithelium, and it is. Not all the cells touch the basement membrane. However, the appearance of this transitional epithelium varies tremendously with the functional state of the bladder. If you're looking at the epithelium surrounding an empty bladder, such as this one, the epithelium is going to look very thick and stratified. If you're looking at the epithelium that lines a full bladder, which is distended, the epithelium looks quite flat. So we're going to take a look at another bladder, which is in the empty state, and then look at one in the full state, and you'll clearly see the transition through which the epithelium goes, hence the name transitional epithelium. The next two images that we're going to look at are of the wall of the bladder, but the wall of the bladder in radically different functional states, mainly empty and full. This is an H&E stained cross-section through the wall of an empty bladder, and here the epithelium is contracted, and it looks like a stratified epithelium, which indeed it is. It's many cell layers thick. Here's the connective tissue beneath the epithelium. Now what I want you to do is hold this image in your mind and compare it to the next one which we're going to show you, which will be the full bladder, in which the epithelium is fully stretched. And here it is, the wall of the full bladder. Hard to believe it's the same tissue, isn't it? In this case, this is the wall of the bladder extending from the luminal surface here to the outside here. The lumen would be in here, and the epithelium has been stretched very, very thin. So its surface is here, and the base of the epithelium is here. Here's the stretched thin nucleus of one of the epithelial cells, another nucleus of another epithelial cell, and from here on down is the connective tissue that forms the wall of the bladder, the connective tissue and the muscles. You can see some muscles stretched tight here. You can see some connective tissue fibers here. You can see a blood vessel here. But the major take-home point is that the epithelium, which is here, of the full bladder, which is stretched, looks completely different from the epithelium of the empty bladder, which is compressed. The epithelium has undergone a profound morphological transition, hence the name transitional epithelium. What I'm going to do now is give you a practical exam to test your newfound knowledge of the histology of epithelial tissue, and the format will be as follows. I will point to something with an arrow, ask you what it is, and give you 10 seconds to make up your mind. At the end of 10 seconds, I'll give you the answer and a brief description of how you can tell what it is. If you need more time than that, just pause the tape for as long as you like. So here we go. Question number 1. Classify the tissue the arrow is pointing to right here. If you said simple columnar epithelium, you're absolutely right. You can tell by the row of nuclei here. Each of these cells is columnar in shape. It's taller than it is wide, and you can tell by the row of nuclei it's a single layer of cells, so it's a simple columnar epithelium. Now, on exactly the same image here, I'm going to move the arrow to a structure right here. And that will be question number 2. What is the arrow pointing to right here? If you said the brush border, you're absolutely right. This is the epithelium that lines a villus of the intestine. It's a simple columnar epithelium. The bottom of the epithelium is here. The top of the epithelium is here. The lumen of the intestine would be right above the arrow. And this dark pink line here is the brush border, which consists of a series of parallel microvilli that greatly amplify the surface area available for absorption on these intestinal absorptive cells. Question number 3. How would you classify this epithelium that extends from here to here? Classify that epithelium. And the answer is, it is a non-keratinized stratified squamous epithelium. Clearly it has many layers, so it's stratified. The upper cells are flat, so it's a stratified squamous epithelium, and you can tell it's non-keratinized because the cells in the upper layer have nuclei. You won't see them in every cell because the nuclei are smaller than the cells, but if you look here and here, for example, you can see nuclei in the upper layer of cells, and whenever you see that in a stratified squamous epithelium, you know you've got a non-keratinized stratified squamous epithelium. Question number 4, classify this epithelium right here. Classify this epithelium right here. And the answer is simple squamous epithelium. This epithelium lines the Bowman's capsule of the nephron of the kidney. And if you notice, you find just a thin strand of cytoplasm here interconnected by profiles of nuclei. This is a single layer of very flat cells, so this is a simple squamous epithelium. Now, in the same image, I'm going to ask you another question. How would you classify this epithelium right here? And the answer is simple cuboidal epithelium. That is a simple cuboidal epithelium that lines one of the proximal convoluted tubules in the kidney. And you can tell it's a simple cuboidal epithelium because the nuclei here indicate there's only a single layer of cells in this epithelium, and the cells are just about as wide as they are high. So there you have a very good example of simple cuboidal epithelium. Question number 6. How would you classify the epithelium the arrow is pointing to here? If you said stratified columnar epithelium, you're absolutely right. And remember, some books will call this a stratified cuboidal epithelium. We're calling it a stratified columnar epithelium, using that as a convention. You can tell it's a stratified epithelium because there are nuclei that lie at many levels. And if you're in some doubt, you can tell it is by virtue of its location, because it's in an excretory duct of a salivary gland. And you can tell it's in a salivary gland because you can look down here and see the secretory acini of, in this case, the parotid gland, which are really quite distinct. Question number 7. How would you classify the epithelium that extends from here clear down to here? Classify that epithelium. That is a keratinized stratified squamous epithelium. You can tell it's a stratified squamous epithelium because it has lots of layers and the top cells are flat. And if you look at any of these cells in here, you won't find nuclei in any of them. And so you can classify it as a keratinized stratified squamous epithelium as opposed to the esophagus that we saw back in question number 3. Question number 8. Classify this epithelium that extends from here to here. And the answer is, it is a ciliated pseudostratified columnar epithelium. Now next, question number 9. What structure is the arrow pointing to right here? In this case, the arrow is pointing to the basement membrane. Now you notice the basement membrane is very well developed in this particular tissue sample. That's because this is from the upper respiratory tract, which characteristically has a large basement membrane. The upper respiratory tract is lined by a pseudostratified ciliated columnar epithelium. You can also call it a ciliated pseudostratified columnar epithelium. And it's characterized by having cells whose nuclei lie at different levels. There's a basal cell, for example, here, whose nucleus is right on the basement membrane. This is the nucleus of a ciliated cell, which is up sort of amidships in the epithelium. And this is the nucleus of a goblet cell here. Remember that each of these cells contacts the basement membrane, and that's why it's really a simple epithelium. It's called a pseudostratified epithelium because the nuclei at different levels give it a false appearance of stratification. And the final question, question number 10. Classify this epithelium that stretches from here to here. What is it? What kind of epithelium is it? And if you said transitional epithelium, you're absolutely right. This is a histological slide of the urinary bladder. The epithelium is the pink region here. The blue region down here is connective tissue that supports the epithelium. And you can see a number of cells here all stacked right one on top of one another. They're almost egg-shaped, and you can tell by their configuration that they belong to transitional epithelium. In order to learn to recognize transitional epithelium, the best thing you can do is look at lots of examples of it and learn strictly by pattern recognition. Well, that ends our final exam. Congratulations for doing so well on it. And if there are any points that are unclear to you, just feel free to go back to the beginning of the tape and look at them again. Thanks for being with us, and we'll say goodbye for now.